I've grown accustomed to experiencing what many outside this field might consider horror. In truth, there is no evil to be found in nature. Though it can be vicious and even frightening, there is wonder in the chaos, a beauty even in the brutality. But sometimes the beauty can be difficult to find, obscured as it is by our basest fears. Nowhere has this dichotomy been more apparent than in my studies of pathogens, many of which transform their hosts into twisted shells of humanity, if any humanity remains at all. Now allow me to continue my documentation of one such study, the interaction between the human vampiric virus and the human zombism virus in a co-infected individual. Note that should this recording be split, the results of the first two phases of this study were recounted in part 1. While these certainly provided invaluable data on their own, it wasn't until the third phase that we observed the most altered progression of symptoms. Indeed, despite my attempts to dispel it, during this phase, only one word could capture what the results elicited in us: horror. In the experimental groups of both prior phases, we began to observe what I considered to be an atypical progression of symptoms, ones that did not align with the established models of either virus. First was the unusual blend of characteristics observed in many subjects, such as the rapid onset of necrosis coupled with the regenerative effects of HVV. This often led to accelerated healing in areas adjacent to necrotic tissue, something I thought unlikely to be explained simply by the additive effects of co-infection. Second, it appeared that many co-infected subjects exhibited unusual immunological responses. For example, the antibodies we normally associate with each respective virus were present, but more commonly, we observed an additional antibody response that appeared to target unique viral epitopes, ones that are not present in either virus alone. I decided to pursue additional genetic testing performed on the viral RNA extracted from tissue samples of co-infected subjects. Astoundingly, sequencing data revealed viral genomes that contained genetic elements from both HVV and HZV. It is my hypothesis that what we were witnessing was, in fact, a form of recombination. As you may already be aware, recombination is a process by which genetic material from two or more different sources is combined to create a new hybrid genome. In the context of viruses, recombination can occur when two or more distinct viruses infect the same host cell and during the replication process exchange genetic material. Because both HZV and HVV share a classification within the Mononegavirales order, it should have been clear that this was a possibility. Instead, the genetic similarities between the two viruses, coupled with their ability to infect the same host cells, were the perfect combination for the emergence of a new recombinant pathogen. Though much more testing will be necessary to determine the exact process, it is my hypothesis that during viral replication, the RDRP enzyme, itself responsible for synthesizing new viral RNA, may have switched templates from one viral genome to another, resulting in the incorporation of genetic material from both viruses into a single chimeric RNA strand, a process known as copy choice recombination. In short, the resulting recombinant virus, which until a more proper name can be determined I have simply called the human vampiric zombism virus, or HVZV, displays a unique combination of genetic elements and functional properties derived from both parent viruses. And though the specific genetic composition of HVZV is likely varied among different recombinant strains, there are some notable consistencies. At the cellular level, once inside the host, HVZV's chimeric replication machinery appears to allow for extraordinarily rapid viral proliferation and spread. The recombinant virus also demonstrates an increased ability to evade host immune responses, primarily due to the acquisition of immunomodulatory genetic elements from both parents. Once the recombinant virus becomes more prevalent, the next phase of our study was clear. We needed a group of subjects who hadn't been exposed to either virus, and would only be infected by the novel HVZV. Once more, we obtained the willing participation of a handful of individuals, pioneers really, who would undergo this unprecedented trial. And it was in this group that we observed the most profound changes yet. The rate of infection in this strain is staggering. While the incubation period averages about 3.5 hours, the expected comatose state was reduced to a state of altered consciousness, more like delirious catatonia. This period routinely spanned 2 to 4 hours. Following this, the virus had spread to nearly every tissue within a matter of hours and had already begun to affect its changes. Internally, in almost all of these individuals, the remodeling that occurred in HVV still took place, but in novel ways. For example, during the coma, the heart ceases to function, presumably due to viral cardiomyopathy, and it appears that it will eventually necrotize completely. As in both HZV and HVV, the skeletal muscle will take over the function of maintaining circulation through rhythmic contractions. In HVZV, these contractions are more controlled than those seen in zombies, resulting in improved circulation, which in turn limits the spread of necrosis. However, these contractions are occasionally spasmodic, causing the subject to tense completely in clear discomfort. The muscles themselves exhibit the proliferation of fast-twitch fibers characteristic of HVV infection, while also recruiting additional muscle fibers in the forearms, hands, and jaws. This results in a subject with the ability to not only move quickly, but exert astonishing grip and bite force. However, I have not observed a significant increase in myoglobin or mitochondrial efficiency, which would explain why, despite being capable of large bursts of speed and power, HVZV victims exhaust relatively rapidly. Now, in a zombic infection, while bite force is also greatly increased, the teeth are not reinforced, leading to their eventual destruction. It appears, however, that during the genetic recombination of HVZV, the vampiric virus's osteogenic and dentinogenic effects that normally apply only to the eye teeth have been somehow modified to include the incisors and, to a lesser extent, premolars. This will certainly require more study to understand fully, but the effect of this change is clear. These modified teeth are a disturbing adaptation for its altered dietary habits. In short, HVZV-infected hosts exhibit a compromised digestive system, which appears to struggle to process and extract nutrients efficiently. Behavioral observations indicate, as in the second phase, that feeding is indiscriminate. And yet, thanks to periodic necrosis of the gastrointestinal lining, the digestive system is not completely adapted to the processing of blood, leading to apparently severe pain and impaired nutrient absorption. Even so, nutrients are absorbed, but the rate of feeding must increase to compensate for its inefficiency. Furthermore, HVZV victims appear to lack the ability to synthesize PCDH-Y, just as in HVV, and must obtain it from their food. As a result of this, recombinant victims are deeply aggressive. Indeed, it appears that the wild frenzy of the zombie is paired with the relatively controlled desperation of the vampire, leading to an extremely effective killing machine, one who is able to hunt with ruthless precision. Despite some level of control, however, it is unlikely that anyone exhibiting this affliction would ever be able to blend into society, as many vampires attempt to do. As I've often stated, the nature of these studies and their associated observations, despite their importance, can take a heavy toll. I consider myself, and even my assistant, to be of relatively stout constitution. It may sound unscientific, but in fact, in watching the subjects as these diseases wrought their insidious and often painful changes, it was clear that anyone who feels nothing at the anguish of another could certainly not be trusted to conduct an experiment objectively. It is my sincere hope that these tests will soon come to an end, and the era of such pernicious pathogens can come to a close. But beyond the physical discomfort exhibited by most of the subjects in this study, it was the changes in cognition that gave me the most pause. Indeed, cognitive functions are profoundly impacted by HVZV. Likely due to the influence of HVV, overall subjects appear to retain a semblance of their pre-infection personalities. However, this is often interspersed with memory lapses, disorientation, and a temporary loss of higher reasoning skills. Similarly, emotional regulation is severely compromised, and infected individuals are highly dangerous and unpredictable. Periods of emotional stability are rare and fleeting, often giving way to intense outbursts of violence. And yet, it is when a prolonged moment of lucidity intersects with awareness of their condition that the most disturbing emotion appears: despair. Fortunately for the victim, though a semblance of balance is maintained for a time, eventually their higher cognitive functions will devolve beyond even this fleeting awareness. The cause of this degradation of cognition appears to be the disruption of synaptic function, in turn caused by the direct infection of the recombinant virus within neuronal cells. The resulting erratic nerve function affects much of the victim's behavior, but interestingly, it does not, at least initially, appear to affect motor control. As in the progenitor HVV, victims of HVZV exhibit enhanced nerve conduction velocity, as well as thicker motor nerve axons, both of which lend to incredibly fast and precise movements and reflexes. As in zombies, I have observed massive stem cell production and the noted absence of glial scarring, both of which lend to astonishing regeneration abilities in damaged nerve cells. In short, if any normal human being were to find themselves stalked by a victim of HVZV, little short of complete destruction could possibly succeed in stopping an attack. But although attacks are powerful and destructive in nearly every case, in HVZV victims, the senses that normally assist with discerning and locating prey are difficult to quantify. For example, the gustatory cortex in many individuals appears to be enlarged, implying the enhanced sensitivity of taste. This, however, is paired with an apparently strong preference for the tastes of blood and living flesh, with varying ability to detect and interpret any other flavor. And as oral tissues deteriorate, taste sensitivity appears to fluctuate rapidly, which only adds to the infected's confusion. Hearing is another sense that is difficult to analyze. It appears that while HVZV victims exhibit enhanced abilities to detect even minute sounds at a distance, some even being drawn to the sound of a beating heart, it seems difficult for the same individuals to locate the sound's source. Indeed, it appears that in each case, asymmetrical enhancements lead to inconsistent sound localization and directional hearing abilities. Olfaction, on the other hand, is greatly increased in effectiveness, as olfactory bulbs and associated cortical processing areas appear hypertrophied in HVZV victims. Though field testing in this study was limited, it can be safely concluded that the scent detection ability in these individuals surpasses that of both vampires and zombies. A truly frightening possibility. The most unpredictable outcome of all, however, had to do with sight. In HVV, victims exhibit quadrochromatic vision, with an additional fourth cone type being tuned to near-infrared. This also appears to occur in victims of HVZV, as does the characteristic formation of tapetum lucidum, which greatly enhances night vision. However, these visual improvements are paired with sluggish pupillary response and reduced function in the visual cortex of the brain, both of which are owed to the influence of HZV. Cataract formation occurred in roughly 40% of individuals as well. The result is a creature with the ability to see a broad spectrum of light and can see well in near darkness, but has difficulty discerning silhouettes and cannot cope well with rapid changes in lighting conditions. Once again, this affects these individuals' ability to hunt with precision, having the counterintuitive effect of making them more dangerous. Interestingly, these deteriorative changes also appear to have reduced the intense reaction produced by intersecting right angles, a trait normally observed in vampires and which I have previously described as the crucifix glitch. Indeed, subjects presented with a cross displayed no adverse reactions, much less the typical seizure-like neuroelectric overload observed in vampires. Lastly, as we observed in the second phase of our study, individuals afflicted by the recombinant virus began to experience chronic inflammation. This, combined with the virus's aggressive replication within nerve tissues, appeared to amplify pain perception. Indeed, even minor injuries seemed to elicit severe reactions, and in 83% of those studied, subjects expressed chronic levels of severe discomfort. This appears to have been alleviated for a time by the influx of dopamine associated with feeding. It is perhaps fortunate for the infected that, given the number of maladaptive traits brought on by this particular virus, their expected lifespan is drastically reduced from what we would expect to see in vampiric individuals. Indeed, it appears that the physiological stress, combined with chronic inflammation and frequent tissue damage, will eventually lead to the failure of vital systems. But more than that, it appears that because of the increased cell turnover resulting from continuous cycles of tissue damage and repair, HVZV actually accelerates telomeric erosion. This occurs so rapidly, in fact, that the increased levels of telomerase brought on by HVV cannot effectively counteract it. The ultimate result is that individuals afflicted with HVZV will likely not exhibit the lengthened lifespan of vampires, but nor do they succumb to the necrotic effects of HZV as rapidly as true zombies. Instead, they are caught in a kind of horrific limbo, highly dangerous, yet equally impaired, their minds and bodies degrading enough to cause pain and unending hunger, yet functional and aware enough to recognize the slipping away of who they once were. If you've listened to this recording to this point, my valued listener, I believe it highly likely that you consider me to be as much of a monster as the poor creatures I've studied. And in some ways, perhaps you're right, but this work must be performed. There's simply no other recourse. This mantle has been thrust upon me, and if the civilized world is to remain blissfully unaware of the threats that face it daily, I could no more refuse it than I could the offer that ushered me into this world of shadow in the first place. My apologies. At this point, you may be wondering, even as I did, given the apparently close relationship of HVV and HZV, and despite their distinct pathologies, is it possible that they descend from a common ancestor? Though I have no direct evidence yet, it is my belief that the answer is yes. Based on the similarities in their genomic structure, cellular entry mechanisms, and zoonotic origins, I propose the existence of a hypothetical progenitor virus, which I have tentatively named the hematophagic mononegavirus, or HMV. The HMV, I believe, originated in an ancient blood-feeding organism, likely an insect or arachnid parasite. The virus would have developed the ability to persist in the host's bloodstream and to be transmitted through the vector's feeding process. Over time, the HMV could have adapted to infect multiple species, including bats and other mammals, through the bites of infected vectors. In these intermediate hosts, it seems likely that the virus underwent further mutations and adaptations, allowing it to better exploit the host's cellular machinery for replication. As the HMV spread to different host species, it likely encountered various unique pressures, possibly in the form of immune responses and competition with other microbes. These pressures could have driven the divergence of HMV into distinct lineages, eventually giving rise to the early form of the human vampiric virus and the human zombism virus. The next step in this research is twofold. First, epidemiological investigations into the geographical distribution and transmission patterns of HVV and HZV, which could potentially reveal common hotspots or overlapping regions where the viruses first emerged. Second, more in-depth genomic sequencing and comparative analyses of HVV and HZV isolates from different hosts and regions, perhaps even non-humans. These could identify shared genetic elements and conserved molecular features, each of which could allude to a common ancestral virus. Until such a time as these additional studies can be conducted, it is difficult to even speculate as to the nature of this progenitor virus in relation to its hosts. It is likely that victims of HMV exhibited acute fever and flu-like symptoms, alterations in behavior such as increased aggression or agitation, heightened senses, especially those related to the detection of blood or potential prey, increased metabolic rate and energy consumption, and perhaps a gradual deterioration of cognitive functions. However, the specific symptoms and their severity would likely vary greatly depending on the host species and the stage of viral evolution. It would be irresponsible of me to confirm any specifics at this time, but suffice it to say that I have wondered if perhaps some of the worldwide legends of the undead could point to a single source, a gap in scientific knowledge that HMV could potentially fill. Ultimately, it has become clear to me that given this recombinant virus's enhanced rate of infection, it is now more important than ever that both HZV and HVV be eradicated, or some kind of cure developed. Even this study was brought to what I consider to be a premature end, as the risks associated with this novel pathogen far outweighed the potential benefits of continued research. The decision to halt the experiment was not made lightly, but the possibility of an unintended release was simply too great. It is my hope that similar studies will be conducted in the future, but with resources and safety measures to which I no longer have access. I will admit that there are times, more frequently now, when I take no joy in my work. There has been intrigue and passion, but the weight of bearing this knowledge has begun to take a toll. The world is still full of mystery, but lately I've begun to wonder if my studies will truly dispel the darkness that conceals them, or if that darkness will simply swallow us whole. This video was made possible by viewers like you. Thank you, and remember, you matter.